Okay, this is an addendum to lectures 18 and 19, the special senses, and this is for the Monday morning Monday morning lecture, since we're not going to have a chance to cover these in class. Remember last time we talked about the special senses, and what makes the special senses special is in fact that they're just located on the head. And so those are senses like smell or olfaction, taste or gustation, as well as hearing, equilibrium, and vision. Hearing and equilibrium are accomplished through the inner ear, specifically something called the vestibulocochlear apparatus, which includes the cochlea, which is used to transduce sound, as well as the vestibular apparatus that transduces things like static equilibrium, dynamic equilibrium. And then finally we're going to talk about vision, which of course involves the eyes. Just a reminder that the majority of the special senses we're talking about use a two-cell or more model. That is, we have one cell that you can see on the left that its only job is to transduce that stimulus, whatever it is, into a graded potential. Graded potentials do not travel down an axon like an action potential does. Instead, in these receptor cells, the graded potential causes the receptor cell to release a little bit of neurotransmitter. This neurotransmitter travels across the synaptic cleft until it gets to our sensory neuron, and if there's an adequate amount of that neurotransmitter, the sensory neuron will form action potentials, which will go to the appropriate sensory region of the cerebral cortex. Okay, so we're going to talk about equilibrium. Equilibrium, that is knowing whether you're right side up or upside down, whether you're accelerating, deceleration, whether you're turning corkscrews like on a roller coaster. These are all part of equilibrium, and they're all sensed by the vestibular apparatus and its associated structures, which are attached to the cochlea. So the types of equilibrium, we'll start out with static equilibrium. That is knowing where you are relative to the force of gravity. So if you're sideways, you know that you're sideways without having your eyes open. If you're upside down, you probably know that as well. And that's because we have certain organs within our vestibular apparatus that tell us about our static equilibrium. Dynamic equilibrium has to do with motion. So if we are rotating, if we are accelerating like in a car, or decelerating very, very quickly, we usually classify this as dynamic equilibrium. Now, interestingly enough, dynamic equilibrium adapts kind of quickly, that as you start to speed off in a car, initially you're aware of that acceleration, but as you come up to static speed, say 50 miles an hour, you're no longer aware of that movement unless you're looking out the window or something like that. So the vestibular apparatus sends information to the medulla oblongata, the pons, and cerebellum via cranial nerve number 8, vestibulocochlear nerve, and specifically the vestibular branch. Okay, so let's take a look at the vestibular apparatus. There's two main parts. First are the otolithic organs. Oto meaning ear, and lith actually means stone. In some animals, the otoliths are very, very large stones that sit in the inner ear and give us information about static equilibrium. Again, whether you're right side up, upside down, or sideways. And they're located in these swellings in the saccule and the utricle, and basically the transduction that happens there is by naked hair cells, and we'll look at those in just a second. On the other hand, the semicircular ducts, which you can see labeled there, remember, give us information on static equilibrium. They are fluid filled, and as you move around, that fluid rushes through those tubes, and you can see that one is horizontal, one is vertical, and one is sort of oblique. And so this is transduced by encapsulated hair cells that are located in the swellings at the base of each of the 3 semicircular canals. And these are called the ampullae. Okay, so let's look at how some of these organs work, our otolithic organs. So here you can see that the otolithic organs have hair cells that are encapsulated in a membrane or gel-like matrix, and above that are relatively small but fairly heavy dense structures called our otoliths. Now, unlike fish, which have these huge big old otoliths, you know how I like to talk about fish, humans just have very microscopic otoliths. But because these are denser than the surrounding tissue, they have mass, and that mass gives them some inertia when you begin to accelerate or decelerate, or even put your head to the side. So you can see in the picture at right that that person has their head right side up, and you can see that the otolithic membrane is resting directly on top of those hair cells. Now, as this guy leans his head forward, the weight of that otolith, or the otoliths in the membrane, actually bends that membrane and bends the hair cells. And by bending the hair cells, they then are going to transduce that into a graded potential and release a little bit of neurotransmitter towards the afferent sensory neurons. And those are the ones that are the red and blue structures that are connected to those hair cells. So remember, this is going to be usually a two-cell model that's involved here. Okay, if we take a look at the ampullae of the semicircular canals, remember the semicircular canals are for dynamic equilibrium. So acceleration, deceleration, as well as rotation. So you can see here that the ampulla itself has a gel-like cupula, and the cupula surrounds those hair cells. There's no otoliths present, but because there's fluid rushing through those semicircular canals, that as you briskly turn your head, that fluid rushes one way or the other and then bends that cupula, like you can see at the picture at right. So the person there is turning their head very quickly to the left, and that causes the fluid in those semicircular canals to move and to bend the cupula in a certain way. And when it bends the cupula, it bends the hair cells. The hair cells begin to secrete neurotransmitter, and that neurotransmitter travels across to the sensory neurons, which generate the action potentials, which are translated to the cerebral cortex, medulla oblongata, and cerebellum. Okay, so now we're going to move on and talk about vision. Humans are definitely very, very visual animals, and that our sense of smell is crap, our sense of taste is not very good, and our hearing's pretty bad as well. But we do have quite excellent vision, maybe not compared to eagles or things like that, but we are at heart predator animals that rely exclusively, or not exclusively, but predominantly on our vision to survive. Okay, so I said before that vision is definitely our dominant sense, that of all the sensory receptors in the body, many of them are in the eye. And if you look at that statistic, that more than half the cerebral cortex is involved some way in vision. So we allocate a much greater proportion of the brain to interpreting stimulus from the eyes than we do to any of the other special senses. Okay, before we go through how the eye works, we're going to talk about the anatomy of the eye. And there's three basic layers of the eye: the fibrous tunic, the vascular tunic, and the retina. The retina is where transduction happens. That's where our photoreceptor cells are located, whereas the fibrous tunic makes up the sort of outside skeleton of the eye. And it's divided into two parts, the sclera and the cornea. You're probably aware that the cornea is the clear part of the eye. It's basically almost a lens on the outside because it does refract some light, whereas the sclera is the white of the eye. And again, we can't see through this sclera, but it's a fibrous connective tissue layer that does provide some structure to the eye. The vascular tunic, remember vascular means vessels, and so this is the area of the eye. It's often pigmented, but contains a lot of blood vessels. The eye is extremely energetically expensive. It needs a lot of glucose, it needs adequate blood pressure, and so forth. And you probably experienced this if you've ever sat up quickly or anything like that, and notice that your visual field kind of restricts itself, or you might even, basically temporarily black out for just a second, and that's because the blood flow to the eye is so important to making it work. So we'll talk about these different parts in just a second: the choroid, the ciliary body, and the iris. Basically, the iris is very important because one of the functions that it does is to restrict the amount of light passing into the eye to what's appropriate. When you walk outside on a nice sunny day, your iris restricts and makes the pupil very, very small to only let a little bit of light in. Whereas if you're sitting in a very dark movie theater or something like that, the iris will expand and make the pupil bigger to let more light in. Okay, so here's the eye, and we're going to go over the parts of it. So on the outside we can see the sclera, that's the white of your eye, makes up part of the fibrous tunic, again made of fibrous connective tissue. You cannot see out of the sclera. And there's our cornea. The cornea is the clear, translucent part of the fibrous tunic on the anterior part of the eye. Light passes through the cornea and is refracted there. So we normally think about the eye and really only having one structure called the lens, but in fact it's important to realize that the cornea itself is a lens of sorts and that it does bend light and refracts it. And for some people, if they are myopic, nearsighted, we can correct their vision with glasses, or we can do that simply by changing the shape of the cornea. So if you know of people that got LASIK surgery or radial keratotomy surgery, oftentimes the structure we're adjusting is the cornea, and by adjusting its shape, we can adjust how it bends or refracts light. Okay, the choroid is part of the vascular tunic. Remember, vascular contains blood vessels, and there's a lot of blood vessels in the eye. You can even see these blood vessels on an ocular exam because the layer over top of the choroid, the retina, is almost translucent. It's very, very thin, nearly transparent membrane. So when you go to the doctor's office, you can see the choroid in a visual exam. And this is what also gives us the red eye when we take photographs in really low light levels, because that iris is open up all the way and that light reflects off the eye, goes through the retina, hits the choroid and bounces back, and therefore in all your pictures, you know, your relatives look like demons because they have these red eyes. Okay, part of the vascular tunic is called the iris, and we already said that the iris's job was to basically serve as an aperture to either let more light in or restrict the amount of light passing into the lens and into the retina. So again, on a very bright day, the iris is going to be restricted, giving us a very small pupil, whereas in very dim lighting, the iris is going to expand, giving us a larger pupil. The pupil itself is just an aperture or just a hole in the iris, and the iris is basically that contraction that happens is through smooth muscle, so it's not a voluntary response, but the iris itself helps us to adjust the size of the pupil. The next layer in back here that we can see is the retina. The retina is the third and internal layer of the eye, and it's only in the posterior, I think 2/3 of the eye. So there's no retina up in the front of the eye, only in the back. And the retina is where the photoreceptor cells are located, and those are the cells that are going to transduce this light stimulus into graded potentials, into neurotransmitters, and they're going to send that neurotransmitter to other cells, and eventually they're going to send that information up to our optic nerve, cranial nerve number 2. Okay, the lens itself is made up primarily of a connective tissue. It's not vascular, but that connective tissue is somewhat flexible, and you can see that there's some suspensory ligaments that are connected to smooth muscle on the choroid. And basically, the lens helps us to accommodate or focus on both near objects and far objects. So depending on whether we're looking near or far, the lens can be stretched out or it can be bunched up. So it just depends on what you're looking at as to what the shape of the lens will be. Up in front of the lens, between the lens and the cornea, is something called the anterior chamber. And the anterior chamber is filled with something called aqueous humor. So it's a very low viscosity fluid that's generated in the choroid of the eye. And this aqueous humor helps to give the eye some of its internal structure and rigidity. If we have too much of it, though, it can cause problems. So probably when you go to the eye doctor, they'll do something called tonometry, where they'll actually generate a puff of air to the front of the eye, or we can actually put a transducer on the cornea and estimate the amount of pressure that's behind there. And what can happen with glaucoma and things like that is that we get too much aqueous humor being generated and not enough drained, and that pressure can damage the retina permanently. And so if you have a higher than normal interocular pressure, they're going to look at putting you on medications to reduce that pressure. Otherwise, you could lose your vision very, very quickly. In the back of the lens is something called vitreous humor. Vitreous means thick or viscous. This is sometimes called the vitreous body. It's basically a big glob of translucent snot-like material. When we look at it in the lab, when we get to cut open the eyeballs, it's really cool because you'll cut this eye open and this big gelatinous blob will fall out. And to me, it looks like those little canned lychee treats that you have in a little cup, and you peel them out and they fall into your hand. So it's almost like a little jellyfish-like structure that's in the back of the eye. But like the aqueous humor, the job of the vitreous humor is to give the eye a pretty symmetrical shape and making it round and non-deformable. Okay, the optic nerve, this is going to be cranial nerve number 2, is connected to the retina, and it receives nerve impulses from the cells in the retina and transmit those through our optic chiasm to the appropriate region of the brain. Okay, so we already talked about interocular pressure and said that that was generated by the aqueous humor, and that glaucoma was an increased interocular pressure, and it results because mainly we don't have proper drainage of the eye, and that this greater than normal pressure can cause the retina to basically die off and eventually cause this progressive blindness. And glaucoma in particular is prevalent within certain ethnic groups, and also your chances for glaucoma gets greater as you age, and there does seem to be somewhat of a genetic component to it. So that's why it's important that you get your interocular pressure measured periodically. Okay, we said that the retina was the sole part of the nervous tunic. It's the posterior 3/4 of the eye. Now in there is something called the optic disc. The optic disc is the exact area in the retina through which the optic nerve exits. Excuse me. There's not actually any photoreceptors in this optic disc area. And in fact, if you look in your lab manual, there's a neat little exercise you can do to visualize your blind spot. You're not aware of it because your brain basically helps ignore the blind spot and fill in the area around it, but you do in fact have a blind spot at your optic disc. Let's see. Detached retina is something that can happen through severe trauma to the head or to the eye. If you've been boxing, which is probably not likely, it can happen. More likely is that we have with some of the elderly patients. If they take a fall and they hit their head, the retina can at least partially detach from the vascular tunic and result in fluid accumulating between those two layers. And initially you might just see a slight reduction in the visual field, but this is something that will progress and progress unless it's treated very rapidly. And so somebody that has had a blow to the back of the head or something like that obviously needs to be evaluated by their eye doctor. Okay, let's take a look at the layers of the retina. The retina is probably the most complex of the sensory structures that we're going to talk about in lecture. If we look at the posterior part of the retina, that is the top of the screen there, you can see something called the pigmented layer. It's made out of epithelial tissue, and the pigment in this layer is basically there in part to reduce any bounce back of light as it goes through the retina. Now the light would be coming from the bottom of the screen. Imagine this person is looking downwards and the light's coming through the retina and then going through all these different neural layers, and then it ends up at the pigmented epithelium. And the pigmented epithelium's purpose is to absorb any excess light and keep it from bouncing through again. The choroid, which is behind it, also has this function, and for that reason it's also black, at least in diurnal animals like ourselves. Those are animals that are active during the day. Okay, the second layer, the one below the pigmented epithelium, is called the neural layer, and this is in fact just an outgrowth of the brain, and these have all the different cells in it that are going to be necessary for transducing and filtering light and making that eventually come out as nerve impulses traveling to the visual areas of the brain. There's three different sublayers. The photoreceptor layer, which is in back. If you see those cells that are located adjacent to The pigmented layer that have the little sort of spiral hats on, those are part of our photoreceptor layer, and the straight ones in there without the little hats are going to be rods, and the other ones are cones. So I'm sure you've heard of rods and cones, and they're used to transduce differences in color vision and also in sort of black and white vision. The blue cell layer that you see below that is the bipolar cell layer, and finally the layer beneath them is the ganglion cell layer. The ganglion cells are the ones that actually generate the action potentials. Okay, so photoreceptors, we said, consist of rods and cones, and they synapse with the bipolar cells. And this is where light is transduced, but the potentials in these cells are going to be different than what you're used to. Normally, when a cell gets stimulated, it becomes more positive, but in this case, the actual potential, the voltage in the cell becomes more negative when the cells are exposed to light. So in the dark, your rods and cones are going to be around negative 40 millivolts, whereas in the light, they're actually going to hyperpolarize to negative 70 millivolts. And what the rod and cone cells do normally is they secrete an inhibitory neurotransmitter. If there's no light around, they secrete this neurotransmitter to the bipolar cell layer and says, basically, don't do anything. But when light is exposed to the retina, they will hyperpolarize, become more negative, and the production of this inhibitory neurotransmitter will stop. In that case, the bipolar cells will become excited, and they will begin to secrete something called excitatory neurotransmitter. And this excitatory neurotransmitter travels over to the ganglion cells, and if there's enough of that excitatory neurotransmitter, we have action potentials generated at the ganglion cell layer. Okay, so here you can see all the different cell layers of the retina properly labeled, and remember that light would be coming from the bottom in this case and going towards the top of the screen. But that the processing, the blue arrow, actually happens in reverse. Processing begins at the rod and cone layer, the photoreceptor layer, proceeds to the bipolar cell layer, and finally the ganglion cell layer, which generate the action potentials. Remember, the retina is a very vascular organ. There's lots of blood vessels that go through there, and so when you do an ocular exam, you'll definitely see plenty of blood vessels in the retina. Okay, cones and rods. Rods are rod-shaped, and along with cones, they're part of our photoreceptor layer of the eye, and they help us to see shades of gray in very, very dim light. You have many more rod cells than you do cones, and again, they're very active in low light situations when cones will not work. Cones require a lot of light in order to work, and they help us transduce very sharp color vision, whereas rods give us a sort of blurry, dim vision that works best at low light levels. So think about when you get up in the morning, and if it's still sort of 5:30, 6:30 in the morning, and it's still dark outside, and it's just beginning to be light, and you look around the room, everything you'll see will be in shades of gray. And if you look at, let's say, the countertop next to your bed, you'll notice the edges look a little fuzzier than what they do in full daylight. And that's because rods don't have the acuity at sharp definition that cones do. So cones are cone-shaped. Those are the cells on our left. We only have about 6 million of those as opposed to 120 million of rods. They're most densely located in the fovea, which is sort of like the focal point of the eye. When you're looking straight at somebody, you're focusing the majority of that light on the fovea. And so this has the greatest density of receptors, specifically cones. On the other hand, if you're going outside to look at something in the night sky, let's say you're looking at a meteor shower or something like that, the rods are not very dense in the fovea, and they tend to be more dense on the external areas. So if you've gone to look for something in the night sky and they say, well, don't look directly at it, look maybe 10 degrees below it, it's because the rods are more densely located or more densely packed in areas away from the fovea. And so your peripheral vision is better for picking up very dim light, say from comets or stars or things like that. Okay, so rods and cones are our photoreceptors, and here's the question: is like, well, how are we able to transduce differences in color using our cones? Now, remember, cones give us color vision, but think of all the different colors you can see. And the answer to this question is that each of the cones has different types of photopigment in there that helps them to transduce things like red and blue and green and so forth. The photopigment is folded into discs in the membranes, and it's degraded as light binds with it. But it's also regenerated at a very, very fast rate. Now, the interesting thing is that if you go out into very, very bright light, think about going to the top of Mauna Kea if there's been a snowstorm, and that light is bouncing back all around you, is that it can overwhelm this photopigment and cause it to break down faster than it can regenerate, so that when you walk inside, you experience kind of what we call snow blindness, and your vision doesn't come back for a few minutes because it's needed for this photopigments to be regenerated. Okay, so we already talked about photopigments, and that all photopigments are involved in transduction of light. There are different types of ospins that are part of the photopigments, and so it says, a cone contains one of 3 different kinds of photopigments, and there are 3 different types of cones, whereas rods contain only one type of photopigment. So they're not giving us the different colors that cones are. Okay, so this is a really important figure. It is actually from a different textbook, and it just basically shows what's going on in our 3 cell layers during both dark and light reactions, and it's very important, something that might show up on an exam. So remember, light would actually be coming from the bottom of the screen, from the entry to the retina, and progressing towards the back of the retina. So if we look at the part of the photo on the left, we can see what happens during a dark reaction. So if there's no light on the retina, what are the photoreceptors doing? Well, the photoreceptors are relatively depolarized, so they actually have a higher resting potential when there's no light. And because there's no light, this graded potential will cause them to release some neurotransmitter, which is inhibitory neurotransmitter. So essentially, these rods are more active in complete darkness, and they tell our photoreceptors via this neurotransmitter, don't do anything. And so the photoreceptors are inhibited by this inhibitory neurotransmitter. In response to that, the photoreceptors don't do anything. They don't release any neurotransmitter towards the ganglion cells, and the ganglion cells don't generate nerve impulses. On the other hand, let's look all the way at the right part of the screen. Here you can see that our retina is being exposed to light. Light is coming in through the retina from the bottom of the screen, going up to our photoreceptors. The photoreceptors, in response to that light, will hyperpolarize. They were at negative 40 millivolts, now they're at negative 70 millivolts. So when they hyperpolarize, their production of that inhibitory neurotransmitter stops. You can see that none of that neurotransmitter is traveling across the bipolar cells. So if we're no longer inhibiting our bipolar cells, those bipolar cells become excited. And because they're excited, they release some excitatory neurotransmitter, which travels towards our ganglion cells. If there's enough of that excitatory neurotransmitter there, the ganglion cells will fire off an action potential, which is represented by those little red hash marks on the optic nerve that you can see. Okay, so here it is, again, dark reaction. Here we have no light penetrating the retina. Okay, in response to that, our rods and cones are going to have a higher resting potential, negative 40 millivolts. Essentially, they're kind of depolarized. And because they are somewhat depolarized, they're going to release an inhibitory neurotransmitter at the synapse of the bipolar cells. Because the bipolar cells are inhibited, they don't do anything. They don't secrete any neurotransmitter towards the ganglion cells. Cells aren't doing anything. They don't fire off any action potentials. Okay, let's look at the light reaction. Okay, light penetrates the retina, goes all the way back to our photoreceptor layer. Okay, the rods and cones at this point hyperpolarize. That is, they were at negative 40, now they go even lower to negative 70 millivolts. And in response to that, they do not secrete that inhibitory neurotransmitter, that they normally secrete in the darkness. And because this neurotransmitter is not secreted, the bipolar cells become excited, and they secrete their excitatory neurotransmitter, which travels across to the ganglion cells, the ones at the bottom of the screen, and the ganglion cells will form action potentials which exit the eyeball via the optic nerve. Okay. These impulses are going to be routed first to the thalamus and then to the primary visual cortex. Remember that the thalamus is sort of a routing station for visual input coming into the brain. And so we go from the eyeball through the optic nerve to the thalamus and finally to the primary visual cortex. Now here's something interesting called convergence. One reason that rods work best in very, very low light levels is that about 600 rods will synapse with a single bipolar cell. And so even though they might be only experiencing low amounts of light, the combination of many, many rods talking to one bipolar cell makes them more efficient at detecting very, very low light levels. So this gives us visual acuity of some sorts at very low light levels, but because there's this convergence, it's almost like a pixelation on an old photograph or an old digital photograph, that the image we see is fuzzy and somewhat pixelated. On the other hand, cone cells synapse directly with a single bipolar cell. And so one cone cell talks to one bipolar cell. And what this means is that we need a lot more light to stimulate the retina in this case, but that when that light is there, that we will have a much finer pixel density with cones than we do with rods. And so this means that cones give us a very fine, detailed vision, very sharp edges and things like that, but it requires that we are seeing very, very high light levels. Okay, things that have to happen in order for us to focus. One is that we have to have refraction of light. Refraction is just the bending of light, and that happens through any optical instrument. When you look through a microscope, remember that there's the optic lens that you look through, the ocular lenses, and there's also the objective lenses. So there we focus or refract the light by getting these lenses closer together or further apart, or getting them closer to the specimen. In the eye, the cornea isn't able to change its shape very much, so the majority of what we call accommodation goes on in the lens, and that the lens does the adjustable bending for the eye. The pupil is also important for focusing. Remember during very, very bright days that your pupil is going to be smaller because the iris is going to constrict, whereas if it's very light outside, where if it's very dark inside, your pupil is going to be much larger because the iris is letting more light in. And the lens, like I said before, does the majority of the accommodation. It's able to change its shape so that light is properly focused on the proper point on the retina. Okay, so here you can see a close-up of what it looks like inside the eye. At the top of the screen we can see our cornea. Remember, cornea is actually a very important organ for refracting light in the eye, but it's not adjustable. And so if you have a misshapen cornea or a misshapen eyeball in general, it can be very hard to focus light. So the cornea does the majority of the refraction, but it's not adjustable. Below that you can see the lens. The lens also participates in refraction, but it's bendable, so it can accommodate, it can help you to focus on things that are very near and far. And the way that it does this is we have special ciliary muscles which are attached to that lens. It can stretch it out like a pancake, or they can release tension and make it ball up like a sphere or a circle. And interestingly enough, the lens does lose its elasticity over time. When we dissect eyeballs this week in lab, you'll get to see that the lens itself in the sheep eyeballs is quite hard, and that's in part because they're old sheep, but also the fixative that we put the eyeballs in makes the lenses somewhat hard. But as you get older, the lens will naturally harden on its own, and this reduces our ability to accommodate to very near objects. And so what you'll see is that older people have to hold objects further away in order to focus on them. Think about how a young child can look at a book or something like that. They can basically read it almost at the tip of their nose. Whereas what is your father doing, or your grandfather, when they read the newspaper? If they don't have their bifocals on, they probably have that held out almost at arm's length. And that's in part because the lens loses its elasticity and its ability to accommodate or focus onto near objects. Okay, this is just a reminder there that the vascular tunic includes the retina, and that the retina's job is to adjust the size of the pupil. And this adjustment also helps us to focus the light somewhat. And so bright light, we've got a very small pupil; dim light, a very large pupil. Okay, we already talked about the lens, so I'm just going to go ahead and skip over this slide because we've already said everything on there. Okay, refraction by the cornea and lens. Remember, refraction is the bending of light, and the two organs that help us to refract light are the cornea and the lens. That almost sounds like a test question. So even though the cornea is not really adjustable, it does do over 75% of the refraction for the eye. And in fact, sometimes if you're not quite focusing on an object, you can slightly change the shape of the cornea and adjust its refraction ability by squinting. Basically, you're using your external eye muscles to change the shape of the eye, and by changing the shape of the eye or squeezing it, we change the shape of the cornea. And so if you're like me and you don't like to wear glasses, you just sit there and squint at things that you can't quite make out. And sometimes that helps you to make out like letters on street signs and stuff like that. Okay, so accommodation is the ability to focus on near and far objects. If you take a look up at top, you can see that that eyeball is viewing, let's say, a tree in the distance that's more than 20 feet away. And at that point, we want a fairly flat lens, and that those light rays are going to be coming in at a nearly parallel angle from that distant object. So they'll come in from the eye. The eyeball doesn't have to do much. It's refracted by the cornea, and then refracted by the lens, and then it's projected onto the fovea, remember, which is the area with the most intense cone density. So it's the area of our focal point in our retina. Okay, now we're going to look at the bottom picture. Here we're looking at something very, very close, maybe just a few inches away. And so as that light comes from that near object, you can see that those light rays themselves are diverging. They're not parallel, but they're starting from a central point and branching outwards. And so here we have to make a nice round lens in order to properly focus that light on the fovea again. And again, as the eyeball ages, that lens is going to lose its ability or lose some of that elasticity, which makes it very hard to focus on near objects. So this ability to focus on things near and far is called accommodation. Okay, so we've already talked about accommodation, and here we talk about different lenses, convex lenses and concave lenses. If you can't remember which one's which, just remember that concave has a concavity, it has a depression away from it. And so what happens is it splits light rays and has them branch outwards, whereas a convex lens tends to focus light on a central point. It moves those light rays close to one another like a triangle. Now, our eyeball doesn't have any concave lenses, but we do have an adjustable convex lens. So the lens in the eye, again, can be stretched thin like a pancake, or it can be allowed to basically form a round ball almost like a sphere. So our lens is a convex lens. Just a reminder what goes on when we're viewing distant and near objects. Distant objects, the light is nearly parallel. The eye doesn't have to do a whole lot of work because it's parallel, and the lens can basically just sit there. We stretch it out a little bit, make it flat like a pancake, and we are able to successfully focus that onto the fovea or the focal point of the retina. And here you can see that what we're actually seeing is upside down, but your brain is able to turn that right side up. Now, viewing a close object, you're looking at something just a few inches away. We have to do a lot more work to bend that light to get it onto the fovea and get it focused. And so here we have the ciliary muscles are helping to bunch the lens into a nice round sphere in order to bend that light and put it on the fovea. Okay, some visual disorders. Many of you probably have contact lenses, probably not a whole lot of glasses these days, but myopia is a nearsightedness where people have a hard time seeing things that are further away. So if you're born in, a few years into grade school, you needed glasses, chances are you're myopic. And this happens in part because the eye itself is enlarged. So look at the top picture, picture A, this is a normal eye. And if you look at picture B, this is a nearsighted or a myopic eye. You can see that that dotted outline in B indicates the proper shape of the eye. But in this case, if you're myopic, you have a longer than normal eye. So what's happening is the light becomes focused before it gets directly to the fovea on the retina. So by the time it gets to the retina, it's out of focus again. So in this case, we give you a concave lens to help focus that on the proper point of the fovea. And so that's at part C. If you can see, this is what it looks like if you have nearsightedness. We just use a concave lens to help branch that light ray out a little bit so that it focuses just at the point on the fovea where the photoreceptors are located. Now, farsightedness, we sometimes call this presbyopia or old man vision. It tends to happen as you age. In fact, you can be both myopic and presbyopic and need bifocals to look at things that are close and far away. They have two different sets of lenses. And so if you are farsighted, basically you need a convex lens to help focus those light rays on their proper point on the fovea. There's also something called astigmatism, which you've probably been tested for at one time or another. This is an irregular refraction of light onto the fovea, and it's caused by irregularities in the curvature of the cornea or of the lens. And so if you have astigmatism, we have special types of lenses which correct for that. Okay, binocular vision. Binocular meaning two. We have two eyeballs, as do most vertebrates. And it's not just because, well, if you lose one, you've got another one, but by having binocular vision, we're able to better gauge how far things are away. So if you take a look at this figure here, you can see the red lines indicate the visual field of the right eye, and the blue lines indicate the visual field of the left eye. And you can see that, wow, they're really, really overlapping, which seems like kind of a waste. Like, wouldn't we want our eyes further apart and able to see more to the sides of our body? Can you think of any animals that might have eyes that are further apart towards the sides of their body? If you're thinking things like horses and deer and things like that, you're on the right track. Because these are prey animals. They're animals that are preyed upon by predators like lions and tigers and things like that. And so they don't have very good depth perception, but they do have a better all-around visual field than we do. We sacrifice some of this 360-degree vision by having our visual fields overlap very tightly. So even though we can't see to the sides of our body as well as a horse can, our visual fields overlap, and this overlap leads us to make better judgments about how far things are away. So we're going to be a better basketball player than a horse or a yak or something like that. And we'll do some neat experiments in lab this coming week to demonstrate the importance of binocular vision and depth perception. Okay, this is just a slide diagramming the different pathways that light takes as it goes through the eyes. It's processed by the retina, and those ganglion cells are the ones generating the nerve impulses. Those nerve impulses travel first to the optic nerve. They cross over to the optic chiasm. And this is interesting. Remember that a lot of the axons from your right eyeball are in fact going to be traveling to the left side of your brain and vice versa. So once we cross this optic chiasm, those nerves are going to go down the optic tract. They're going to be routed to the thalamus and then to the primary visual cortex of the brain, which is located inexplicably way back on the occipital lobe. You would think that that processing would go on closer to the eyes, but it's as far away as can be and at the occipital lobe. In fact, if you receive a blow to the back of the head, this can cause temporary blindness and can interfere with vision.